guys, welcome back. Today what we're going to be looking at is actually assembling all of the things that we've talked about in the previous 3 videos into an entire genetic map for the whole genome and using that to find genes that we're interested in. So genetic mapping can mean a few different things when you talk to a geneticist. So one of the things that it can actually mean is something like creating a genetic map like we did when we did the 2- and 3-point test crosses in the last couple of videos. Another thing that you could be talking about is using that map to figure out where important genes are. So last time we focused on the first part of this, trying to create these maps, actually get them sorted out. What are these distances? And today we're going to be using maps that we can create to figure out where important genes are that are going to give us the phenotypes we're interested in. So focus is on finding cool genes today. So to get started with the kind of mapping when we're looking for something, we need 2 things. The first of these is markers. Okay, we've been talking about markers quite a bit, and we're going to review them a little bit today. And the second thing that we're going to need to make all this work is an interesting phenotype. So again, starting out with our review of markers, important to think about markers as, again, being things like CODIS markers, for example, PCR-based markers, things like that. So remembering markers, again, relating this back to our physical map of Hyrule, markers are the places that you can see. So we know, because we've watched the previous 3 videos, plus at least know a little bit about Hyrule, know that this is not the only 2 places that we can go. We just know we can only see those parts. When we think about this in relation to chromosomes, again, there's going to be 2 chromosomes under here, but these markers are the only places that we can see. We can see the genotype, where these 2 chromosomes, whatever it is going to be related to this darker red, which may be wild type, darker green, maybe wild type, lighter green and red. Okay, and these other 2, we know that it is an alternative allele. But if we can see the whole thing, then we know what's going on. So again, markers, we're going to be something like a gel-based assay. We're going to be able to say, oh, this is going to be the darker red, the lighter red, and where our heterozygote's going to be. Okay, so I just pulled this gel from somewhere on the internet. It doesn't really have a lot of meaning, but just to give you an example. Or alternatively, something like fragment analysis. This looks like CODIS markers to me down here. So again, those ones used in the FBI. We're able to tell size differences and kind of put this together. So just kind of keeping in mind the sort of data you're actually generating, because you can't actually look at the chromosomes and tell them apart by color. Okay, that's just for our convenience now. So in our review of markers, I think it's important to think about, how do you actually get these markers? Like, who came up with the CODIS markers, that kind of thing. Okay, so we're not going to talk about who came up with the CODIS markers per se, but think about those as being kind of single one-off markers. So each CODIS marker is looking at only one location at a time. Okay, so each of these kind of looking at an individual spot. They're often based off of a PCR type of assay, variants on that. Sometimes they're based off of a single nucleotide that's different between 2 alleles. Maybe it's based off of a deletion or something like that, just a small thing that we can check, either size-based assay, maybe a sequencing-based assay, but something that's going to show us one spot. Okay, and of course you could combine these. You can have multiple of this type of marker and visualize them by size. The other way to get markers is to generate a whole lot at once by using a variation on whole genome sequencing. So being able to gather a lot of information, maybe not the physical sequence, but being able to really generate a lot of information. Okay, and that might seem a little bit weird to say after we just spent a whole previous video figuring out indirect genetic distances between things. Like, why did we do that when we could have just figured out the physical? Okay, this is going to be important, again, not just for getting the layout of the chromosome. This time we're actually going to be looking for important genes. So that's going to take a little bit more work, and we're going to need a lot of markers. Okay, so we're still getting up to that. All right, and again, because we're going to have a lot of markers, we can look at a lot of locations at once. So you can think of those single markers. Again, you can combine them. It's maybe looking at 1 or 2 places at once. But when we start adding a lot of markers, okay, we can see a lot more places. And I kind of got tired of making circles. There should probably be more for this to work well. But when you have a lot of these markers, you can see that you can start to get the real lay of the land. You can start to actually see kind of at least vaguely what's going on. If we relate that back to the chromosome, again, we started out with a situation where we only had 2 markers at whatever location this green area is going to be, and maybe a second gene that's going to have whatever this red is. If we had more markers, this would be the information that we could see, okay? So we'd be able to get a lot better lay of the land. We may not know exactly where recombination occurred, but we'd have a lot better idea than just, you know, somewhere between these two, okay? So first thing we needed was markers. We're going to come back to that, circle back around to it at the end when we kind of combine it with our other principles we're going to be looking at. But the second thing we needed was an interesting phenotype. So what is an interesting phenotype? Like, what in the heck is that? Okay, so technically an interesting phenotype is anything that's measurably different that you think is interesting. So this can vary from person to person, okay? And if it's interesting enough to you that you really want to find out what's going on, that's your interesting phenotype, okay? For me, that interesting phenotype is differences in flowering time in different types of sorghum, which is actually the 5th most economically important cereal crop worldwide, if you weren't aware. Okay, it's related to corn, and it is economically important in a lot of places. Okay, so we were concerned with the late and early flowering nature of these plants, because plants that flower early, they're going to stay kind of short, maybe about waist high on an adult. Okay, and these are really good for like making grain, going to be able to harvest these really easily. Whereas these late flowering ones, they actually don't stop growing until they are going to eventually start to make seed. But at this point, they could keep growing forever. Okay, so these we were more interested in when we're thinking about producing a lot of plant material to ferment into ethanol for bioenergy. So that was kind of the goal. You can get a lot of grain with the first one, bioenergy with the second one, figuring out what's going on so that we can get what we wanted out of these plants. Okay, so that's the setup. Now we got to think about, as a geneticist, the background for figuring out effectively what is the difference between these 2 plants. So that's the goal today. What genetically causes this difference? Okay, so again, as geneticists, there's a lot of backstory to all of this stuff. So we have to do a little bit of it just so that we kind of understand what happens at the end. Okay, so when we start out wanting to study this, we know that we've got an early flowering individual and a late flowering individual. I'm going to give the early flowering one pink DNA just so we can see it visually, blue DNA for a late flowering one. Notice it's pink all along, both homologous chromosomes are pink. These are what you would think of like as commercial inbred lines, where they are, you know, pretty much homogeneous along the length of it. Okay, this one for simplicity, we're just going to keep these homogeneous as well. And so what we do is we do a cross, okay, send some pollen, like a very gentle love letter to our other plant, okay? So there we go, some pollen has left, and over time, we're going to end up making offspring, okay? So this is what's referred to as the F1 population. And it's important to note here that this population is going to show the dominant phenotype. In this case, that's the late flowering state. So all of these F1s are going to look kind of late flowering, okay? They're going to be, importantly, heterozygous. So they are going to have these little heterozygous chromosomes floating around. That's going to be important, okay? So let's take some of these individuals. Let's just zoom in on some of them. Okay, take their little heterozygous cells. Okay, and what we can do is actually start to create a scenario where recombination will start to occur between different chromosomes in these individuals. Okay, for now I'm just showing you one chromosome out of the however many this plant would have. Okay, let's just say 10. All right, so this is just one of the 10, but you can see what's going on. We would either self this, okay, it's okay to have self-love, self-care is a thing, okay, and we can see how that's going to work, or alternatively, depending on what you're trying to do, you can cross this to other individuals in this F1 population, okay, so you can self or cross this, and what's going to happen here is we're going to see some recombination occurring, okay, so hopefully in different places, because again, this is all random. We're going to have some crossing over events in different areas. So we're not concerned about the markers yet. We're just trying to get these recombination events to happen. Okay, so 1 or 2 per chromosome is not going to be enough. So what we have to do is continue this scheme, produce more, okay? In our F2, we'll start to see that recessive one kind of plinko out, but we're going to end up with more recombination. You can see that we start to get a mix of pink and blue DNA, continuing the scheme on and on. Okay, so yes, as a geneticist, this actually does take a long time. In sorghum time, probably each one of these crosses takes ultimately about a year or so to kind of get going. So this is a long time. You end up with a lot of plants, thousands of plants, and you end up eventually, when you're satisfied, maybe like 6 or 10 generations later, you're satisfied that enough recombination has occurred. You end up with the FN population, N being the number of times you crossed it, not like the FN population, like you're tired of this already. Okay, so we just have a lot of plants that a lot of recombination has occurred in. And so if we kind of zoom in, let's take one of those guys, okay, and we can look at all their chromosomes. Hopefully all of them look like a good mixture of pink and blue, probably a little bit more than this, but I got tired of making pink and blue areas. Okay, so they would be a good mix and match. Okay, so now let's zoom back out and take 10 of those individuals from our FN population, where we've probably let these go maybe like 6 or 10 generations again. So we've let them cross for quite a bit to get all this recombination. And again, these plants will have more than one chromosome. And if we think about chromosomes as being kind of one map that we can explore, kind of different worlds, okay, obviously we would be wanting to think about finding this in the entire genome. Okay, that would be the whole thing. But for simplicity, let's just take one of those chromosomes, or kind of corresponding to one of these maps. Obviously, we're going to stick with Hyrule because I've already put a lot of prep work into that. Okay, so let's pretend this is chromosome 5. I don't know, I just made that up. So here is going to be chromosome 5 from 10 different plants. But wait, don't peek. We don't know what's on these chromosomes yet because we don't know anything about the markers. We haven't made any markers. We haven't tried to tell. We don't actually know. Okay. But what we can do is remember what the phenotypes of these plants were and start to sort them out. So we had an early flowering parent and a late flowering parent. So we can categorize these offspring as having a parental phenotype or, you know, one or the other. And so we can start to let these guys sort out. So that's not recombination. That's just us pushing all the early flowering ones to the left and all the late flowering ones to the right to sort out later. So we've gotten kind of organized by phenotype. Again, all of the plants, we extracted DNA from them. All of those plants, the DNA on the left came from plants that's early flowering. The DNA on the right is going to come from late flowering plants. Okay, so again, we don't know anything about these chromosomes yet. Okay, so we're getting there. We can find out with markers. Okay, so if we check a few markers, okay, we might see something like this. Again, the more markers you have, the more that you can see. Okay, so you can see a lot of stuff here. But our question now, we've got kind of a visual on what's happening. We've got phenotype sorted out. What the heck are we looking for? Okay, so in its most basic sense, what we are looking for is the place where the DNA is going to back up the phenotype. So in other words, in this example, what we're looking for is a place where pink DNA is always present across the board in all the early flowering plants, so a spot where that's true. And then that same area, we've got to have blue DNA in the late flowering plants. What that means is that whatever gene was causing late flowering, early flowering, the flowering time, if it's always from the blue parent and you see late, always from the pink parent when you see early, what that means is that that gene is in there somewhere. Okay, so take a second, pause that video, and see if you can find where this gene is. Okay, welcome back. Did you guys find it? Hopefully you were able to see that this is going to be right in this region. Because again, what we're looking for is a place where the DNA that came from our original early flowering parent is always going to be present in this area. So the gene that causes it's in here, and you see consistent genotype to phenotype. Anything that's late, we're going to have DNA that came from the late flowering parent. That means that the gene is in here somewhere. Okay, so if we're doing this in real life, we're not going to do it this way. We're not going to do it visually. But what we're going to do instead is, again, take these markers. They're the places that we can see, but again, these are going to be sequence differences. They're going to be places that are different, okay, that we can look at it in a gel, or size differences. We can say allele 1 or 2, which one do I have here? Okay, and then we can put all that information in with the phenotype into programs like QTL Cartographer, or some things that are going to help us map out this a little bit more. And what you end up with is data that looks kind of like this, okay? So this is actually pretty easy to interpret. What we have here, okay, and this is a cross that I set up, so QTL, this is a quantitative trait locus. What that means in unjargony terms, if we just de-jargon this a little bit, that is going to be the place where the genotype is going to back up the phenotype. So that's that genomic area where we always have pink DNA in the early flowering plant. We're going to always have blue DNA in the late flowering plant. MA1 is what, this is just a locus. That's what I call that region, because that region's too large for me to really like say what gene it exactly is. I'll just give it a nickname for now. And these were the parents' names, okay? Just like if their names were like, I don't know, like Alicia and Greg. Okay, so it doesn't matter. These are just the plant names that we gave them. In this case, this was an F3, so we didn't really do too much with this, but that's okay. Here, what we can see across the bottom, the x-axis, these physical bins are actually chromosomes. So this is like what's going on chromosome 1, 2, 3, 4, 5. And what's going on the y-axis is what's called a LOD, or log of odds score. The important thing to know about this is going to help us have kind of assign some statistical significance to what's going on. Okay, so the threshold that we're looking at here, this is going to be kind of a threshold that we set. Anything above that is likely to be an area that caused a phenotype. So what we're saying with this log of odds scores is that this is going to be an area that is responsible for the phenotype that we see. Okay, again, flowering time is what we're looking for. Okay, and with the data we put in, this says, hey, something that's responsible for flowering is going to be present here. Okay, so I know there are multiple peaks. What this means is that each of these peaks could be playing some role in flowering time. The one that I was particularly interested in at that time when I made this is going to be maturity locus 1. Again, locus just because this is too large of an area. This is usually somewhere about, I don't know, maybe like 500,000 nucleotides. It could be a little more, a little less. Okay, but this is going to be the area where a gene is that's going to be responsible for that phenotype. Okay, this is my population size. Okay, so this is not that large of a population size, but that's why I got such a rough map with such a large region. Okay, so when we relate this back to an actual idea of a physical map, again, when we started this whole journey, we talked about markers as being places you can see. Some of them were just passing through, like CODIS markers. We don't necessarily care about the CODIS marker itself, but what we care about is the result we can see from it. So we called these genes that we care about, the ones that are going to be underlying the phenotype that we've been studying, destination genes are more like those Hyrule-Castle genes. So I think that it's always a good idea when we finish this kind of thing to kind of explore the sort of data that you'll really see when you get out in the world, reading papers. What is a map going to look like? Okay, so throughout this journey, we've created a map, and then we've located something we were interested on it. A lot of times, what you'll see in papers is going to be kind of the initial creation of that map, And then you'll see something like the log of odd score plot that I showed you on the previous slide. But when you do see papers that are going to kind of delineate out these genetic maps, what you'll often see is some kind of mess like this. This looks absolutely awful, like your brain just shuts down because what is all of this? Okay, but I want to break this down because you guys now can understand. We've gone through enough stuff that you can understand this. This isn't too bad. So this is the actual chromosome we're looking at, okay? In principle, there would be however many more of these maps that there are going to be number of chromosomes in that organism. Everything on the right-hand side, these are markers. These are the places that they checked. These are the places that you can see. These are marker locations. This is what you can see. Okay, they often have really terrible names like this. A lot of times these refer to either like what the sequence difference is, or they might refer to the type of allele difference. Is this going to be an insertion, a deletion? Is it going to be a single nucleotide change? So these have like kind of a coded meaning, but again, they don't, you can call it like marker 1. It's the same thing. It's a marker location. Okay, so those are just the places that were checked along the chromosome. Okay, on the left-hand side, we have genetic distances, which you can see are kind of additive as you go. So we're adding these up, okay, 14 being this distance between this original marker and here, 14.4. Okay, again, we're actually just another 14.4 away from this one. Here, you know, what is this, like 12 away, 12 again, here's 5 map units away. So we're kind of adding these up as we go. Yes, this does add to more than 50%, but that's okay, because what we were talking about with that idea in those first couple of videos where you can't add up to more than 50%, we're not talking about between necessarily individual markers. Here we're kind of making like an additive map. So that's okay for right now. All right, but I just want you guys to be aware when you do go off into the real world and you have to start looking at literature that's going to tell you about maps, a lot of times you'll see something like this. This is just, we created a map. Or alternatively, you might see things like here, we found something on a map that was previously created. That's where you get those LOD score types of things. Okay, so in conclusion, we've actually put all of this together now. All right, we've put together all the marker work we've been working on all semester. We have put together this idea of expectations for Mendelian ratios. We've looked at linkage, which is really important. We've done a 2-point and a 3-point test cross. We've generated a map ourselves using those 3 plant genes in the last video, and now we've actually used kind of that information. We haven't physically made a map of the whole chromosome, but in the background there'll be a map of the whole chromosome with all of these markers. Put this into a program, and now we're able to actually see this. So our mapping, we're being able to put together these genes in order like that juice color shape from last time. We're able to take advantage of recombination to do this. That's why we have to continue selfing those plants in this breeding scheme to create those instances of recombination. Once we do this, we can compare physical and genetic properties of the chromosomes. We can see what's actually going to be happening. We can look at the phenotype compared to the genotype, and then we can see that these maps help us find out what genes are going to be responsible for what we observe phenotypically. So this is important not just in plants, but in all types of things. So in medicine, people do what's called association mapping to look for associations between disease phenotypes like heart disease and genetics. So we're not going to be breeding humans however we want, but you can look at a large spread of different people, categorize them into kind of bins like, do you have this disease, do you not, and then look at all across their genome and see what's similar. So we've done a lot of things with this. This is our last video in the genetic mapping section. So I'll go ahead and end with our biology joke of the day.